by nutrition. So if we observe all the organisms are needed food for their repair and growth. Yes sir. All the organisms are needed food for their growth and repair. So here we have to discuss nutrition. What is mean by nutrition? Nutrition means ingestion of food ingestion of food into our body. So what will happen after the ingestion? It has to be get digested. Then after absorption, then after defecation will be takes place. So if we observe what is meant by nutrition, there is a definition. 2 marks question. If they ask 2 marks question, nutrition. What is mean by nutrition? Procurement of food of an organism is called nutrition. Procurement of food of an organism is called nutrition, right? So if you observe, nutrition is two types: heterotrophic nutrition, autotrophic nutrition. Autotrophic and heterotrophic. Based on the procurement of food of organisms, these are two types. Organisms are divided into two types. First one is what? Autotrophic nutrition. What is mean by this autotrophic nutrition? If auto means what? Self. Auto means what? Self. If the organism is preparing its own food, those are called autotrophs. What are those called? Autotrophs. This type of nutrition is called autotrophic nutrition. For example, plants and algae. Plants and algae are the example for autotrophic nutrition. Next, opposite to it, heterotrophic nutrition. Hetero means what? Depending on others. Why they are depending on others? The organisms which are depending on others for their food needs. Why? Because they cannot prepare their own food, right? So these type of organisms are called heterotrophs. Heterotrophs. And the nutrition is called heterotrophic nutrition, right? So if we observe here, autotrophic nutrition means what? Plants and algae are capable to produce their own food. That is because of green color pigment. What is the green color pigment present in the plants? Chlorophyll. Chlorophyll is present. Only because of this green color pigment, chlorophyll, the plants and algae are called as autotrophs. Okay now, what is the necessity or what is the importance of this green color pigment, chlorophyll? Only this green color pigment is nothing but chlorophyll is capable to trap the solar energy and it convert the solar energy into chemical energy. Only this chlorophyll can capable to trap the solar energy into chemical energy. So that solar energy also capable to trap in the form of photons. What is mean by photons? Energy. What energy? Light energy in the form of photons. And this solar energy is converted to chemical energy. Solar energy is converted to chemical energy. So solar energy is nothing but photons. Okay. Chemical energy is what? Carbohydrates. CH2O. What it is? Carbohydrates. Okay. Sometimes carbohydrates are going to convert it to starch in the dark phase or in the light independent reaction. Right? Alright. If you what is mean by photosynthesis? Photosynthesis is a life process which is done by plants. So plants are capable to convert the solar energy into chemical energy. So what is that solar energy they are using? So that solar energy is coming from sunlight. So only the light source is what? Sunlight. Right. If you see, plants are taking carbon dioxide from the external environment and also they are absorbing water through their roots in the presence of sunlight or light source. They have chlorophyll in them. and they are converting that solar energy into chemical energy, nothing but CH2O, again plus H2O, and also plus oxygen gas is evolved. Okay, this oxygen gas is used as respirable gas. Whatever the gas we are taking inside in the process of respiration, that is what oxygen gas is used as respirable gas. Next, so what they are taking? Inorganic substances. What are the inorganic substances? Carbon. So plants are taking simple compounds and preparing complex compounds like carbohydrates, right? So these carbohydrates and water and also oxygen is released as end products during the photosynthesis process. Here we have one question. What is photosynthesis? Plants are capable to perform a life activity to produce food material for us. In the presence of light, they have chlorophyll in them. What they are taking? Carbon dioxide they are absorbing from the external environment, and water they are collecting from the roots with the help of roots from the soil, and in the presence of light they are preparing complex substances or molecules like carbohydrates, water molecule and oxygen. So here CB Vanil explained about it in this way. What? To form 1 mole of carbohydrate, to form 1 molecule of carbohydrate or 1 mole of carbohydrate, it needs 1 mole of carbon dioxide and 2 moles of water. 2 moles of water to form 1 carbohydrate and also 1 molecule of water, and oxygen gas is evolved. So this is simple equation for photosynthesis. If you want to make it as a complex, is nothing but balanced equation. So if you know 6 table, it is 6 ones are 6. So if you write 6 here, here you have to write 12. 6 CO2 plus 12 H2O, C6 H12O6, that is nothing but glucose, and 6 H2O and also 6 O2. So this is the balanced equation for photosynthesis. The same CB Vanil explained. So 6 CO2 plus 12 H2O gives rise in the presence of sunlight. They have chlorophyll, and we are going to get glucose. What it is called as glucose, water molecule and oxygen is going to release, right? So first, before this, he used H2S, hydrogen sulfate instead of water. So by that time the same gas was evolved. What is that gas? H2S, hydrogen sulfate gas was evolved when he observed in the purple sulfur bacteria. So he used purple sulfur bacteria. So whenever he used purple sulfur bacteria, what happened? Hydrogen sulfate gas was evolved instead of water. What he used? Purple instead of oxygen gas, sulfur gas was evolved. This is about the photosynthesis equation. So here we can say one thing. What it is? Only plants are capable to prepare their own food, and for themselves and for us also. So that is why plants are called as universal food providers, right? Von Helmont observed that. What he observed? He conducted experiment and he proved the importance of water in photosynthesis process. Presence of starch. What is meant by starch? One of a type of carbohydrate. Right. So we are going to check whether the leaves are contains starch or not. So that is only presence of starch. First activity in our textbook, very easy and important. So if you are going to write any activity in exam, so you should write aim, materials required, procedure, observation and conclusion. These 5 steps should be there. Then only you will get full marks, 6 marks, easy. So sometimes they may ask only procedure and materials required. So even though, so just you write aim. What is the aim? To test the presence of starch in leaf. To test the presence of starch. Where we are checking? In leaf. Right. So every activity needs some materials. So that is called materials required. So what materials we required? By seeing this we can understand. What? Once see. Materials required. What? First one, beaker. Then after, test tube. Then we are taking leaf, no? So healthy leaf. Then after, water, methylated spirit. Next, asbestos gas. Next, tripod stand. Next, Bunsen burner. Bunsen burner. So these are the materials we required. Not only this, if you see petri dish, another name of petri dish, what? China dish also we can write. China dish. Laboratory brush, dropper, iodine solution, so these are the materials we required to perform this activity. So first one, what is the procedure? First of all, we have to take a leaf in a beaker and boil it for 2 minutes. Then what will happen? The leaf will be get softened to insert into the test tube. Okay, now that's why we have to boil the leaf for 2 minutes in water. Then after, whenever it is get softened, then we have to insert the leaf in a test tube which contains methylated spirit. So inside this test tube, what we have? Methylated spirit is there, no? Why we are taking methylated spirit? To remove the chlorophyll in it. To remove the chlorophyll. Just now we discussed green color pigment in the plant. What is chlorophyll? So that should be removed. Then only we can test the starch. So that is why. Next, we have to keep the test tube in the beaker with the help of Bunsen burner. It has to be get boiled up to 5 minutes. Okay. Then take out the leaf from the test tube with the help of laboratory brush. So we have brush. Why? Because it is acid. Do not touch it with your hands. Next. So if you observe, if you take out the leaf in a petri dish, okay, na? Spread it uniformly. Spread it uniformly. Nothing but without any foldings. Now add what we have to add? Iodine solution, drop wise. Okay, 2 to 3 drops are enough. So add 2 to 3 drops of iodine solution to it. So up to here it is procedure. Okay. Next, observation. What is the observation? Before adding the iodine solution, it is in which color? Pale yellow color, or else colorless. The leaf will be colorless. Why? Because chlorophyll was removed. But now when you added this iodine solution to it, what will happen? This will be changed to bluish black color. Bluish black color. So this is your observation. So whenever we added 2 to 3 drops of iodine solution to it, then it turns to bluish black color. So it will be changed. Next, conclusion. This bluish black color confirms us presence of starch in the leaf. Is was confirmed. So starch is present in the leaf. That was confirmed by us.